Today we have the pleasure to have as a speaker Amir Muawiya. He is a doctoral researcher at the University of Tuscia in Italy, but he has been also working with us at the Institute for Sustainable Agriculture in Córdoba. So today he will present a part of his PhD that is focuses mainly in development of sustainable endotherapy treatments against Xylella fastidiosa. Specifically, he will talk about the mechanistic efficacy of cellulose nanocrystals. So Amir, the floor is yours. You have the time you consider is needed for your presentation. Thank you.
Thank you so much, and good afternoon, everyone. I would like to express my gratitude to the organizers of this seminar series to give me the opportunity to share the findings of our research. And today we are going to discuss a novel sustainable endotherapy solution against Xylella fastidiosa, and that is cellulose nanocrystals. So, uh, I'm going to keep the introduction to Xylella fastidiosa very brief for this audience. However, some colleagues may have joined which are unfamiliar with the Xylella fastidiosa. So basically, Xylella fastidiosa is a xylem-limited gram-negative bacterium which creates the biofilm inside the vascular bundle, especially the xylem vessels of the plants. It clogs the xylem vessel, the biofilm clogs the xylem vessel, and the plant dies of the thirst due to the unavailability of the water molecules to the different parts of the plants. So this disease is transmitted through the sap-sucking insects, and critically, there are more than 500 plant species which are susceptible to this pathogen. And only across the Europe, the annual losses are more than 1 billion euros every year, primarily from the viticulture and the olive oil industry. And most importantly, so far there is no curative treatment for this pathogen. The only option to manage this disease is to uproot the plants and burn them. So this is the um, this is the situation, and the European Union mandates very strict quarantine measures to prevent the spread of this pathogen. However, since the first appearance in 2013 in Apulia, it has been spreading across the southern Europe and among different hosts. So, the barriers to tackle the Xylella fastidiosa, as we all know, that the vascular inaccessibility using the conventional methods is one of the problem that we are facing to tackle the Xylella fastidiosa. And then the biofilm exopolysaccharide matrix, it resists the antimicrobial compounds to reach the active, active infection site, active colonization site. And then there could be another viable but non-culturable state of the bacterial cells. Am I audible? Am I audible?
We can see the slides.
Yes.
Yes. Mhm. No problem.
Okay, thank you so much.
We can listen.
So,
Yeah.
okay, thank you. So, the culturability loss in response to some antimicrobial compounds may not be equal to the elimination of the bacterial cells, which means there could be some latent population which can resurge and create a different situation at the end point of not only the experiment but also in the field conditions. So, here we propose a nanomaterial candidate, that is cellulose nanocrystals developed from the waste wood pulp due to its high aspect ratio, because of the rod shaped geometry. Its width is 2 to 5 nanometer and length is about 100 nanometers. It provides a high aspect ratio which can create physical nanobarrier network within this negative charge because of sulfate ester group. It provides a very stable colloidal suspension into the aqueous medium. which may electrostatically interact with the bacterial envelope, bacterial cell membrane. This is a physical mechanism. There is no resistance risk of developing the resistance against this solution by Xylella fastidiosa. It's biodegradable, non-phytotoxic, as confirmed in this and many other studies. So, based on these rationales, we propose a hypothesis that if we inject cellulose nanocrystals directly into the xylem vessels, they may form a physical nanobarrier against Xylella fastidiosa, which may restrict the bacterial translocation across the plant stems and the different tissues. Also, it may induce a growth arrest, and ultimately, the plant can prevent the symptoms. So our experimental design is three-tier. First of all, we studied in vitro the efficacy of cellulose nanocrystals against, I forgot to mention in the beginning, we studied two subspecies of Xylella fastidiosa. One is Pauca strain Cordero, which was responsible for olive quick decline syndrome, and the other was subspecies fastidiosa strain Temecula 1, which is responsible for Pierce disease in grapevine. So first of all, we assessed the uh, behavior of Xylella fastidiosa in presence of cellulose nanocrystals under in vitro conditions in terms of the time kill kinetics, observing the CFUs, colony forming units developed over the period of time, and usually we maintained studied the turbidity for assessing the biomass growth of bacterial cells in PD3 growth, and then we measured the biofilm using the crystal violet assay, and last but not least, we observed the viability of bacterial cells using the PMA XX dye to discriminate the live and dead cells. Based on these in vitro evaluations, we moved toward the in vivo implantas studies, and first of all, we studied the phytotoxicity in tobacco host model plant using the mesophyll assay for different concentrations ranging from 0.01 to 1% weight by volume cellulose nanocrystal suspension in sterile water. And the biocompatibility was initially measured as Leyen scoring and then confirmed throughout the study in terms of different parameters. So when we found that this is a non-phytotoxic and can be suitable candidate for the vascular applications, we moved in vivo and using the tobacco as a host model uh, or CBD design. So first of all, we precisely inoculated the tobacco plants on three sides at the one side of stem near to the base using a Harvard apparatus microinjection pump. And then we treated those infected plants with cellulose nanocrystals, 1% weight by volume suspension at 3 and 7 days post inoculation at proximal and distal to the inoculation sites. We observed the plants for 60 days of incubation. For 60 days, maintaining in the controlled greenhouse conditions in terms of and disease progression, using the handheld devices like DualX and Polypen to observe the non-destructively the response to the bacterial infection. And then we observed three different stem parts below the infection point, above the infection point, and at the top of plant to see the bacterial load at different locations to see if our hypothesis of nanobarrier is valid. So first of all, the in vitro results. Here we have observed a very clear, distinct dose-dependent suppression of the bacterial growth in terms of the colony-forming units on the agar media. So the denser the green color here is, the more the cellulose nanocrystals concentration, and the dark green line, this one represents the 1% weight by volume. And we can see that in case of Cordero strain, the growth was completely reduced to zero at 48 hours. while also for the Temecula 1, we observed the reduction of 5 log units at the end of experiment. So both the strains represented a very clear dose-dependent response, even though the first three treatments, the least concentrations were not statistically different in terms of the growth. However, the top two treatments, 0.5% and 1% weight by volume, shows that the 0.5% is a threshold level. So we continued with 1% in our in vivo observations. So the growth kinetics were further a very sophisticated instrument at Institute of Sustainable Agriculture, which observes the absorbance at every 6 hours of interval throughout the incubation period. So these different colors represent different treatments, and the yellow and the purple one represents the top two treatments, 0.5% and 0.1%. We can see there is a beautiful and very clear growth reduction throughout the period of time, and which is further verified by the total growth calculation. However, the interesting thing is the increase in the cellulose nanocrystals also delayed the log phase of the bacterial growth. In case of the control treatments without any modification into the PD3 growth, the curve was very sharp at 20 hours. However, for the higher treatments, it was extended up to almost 80 or 90 hours of the incubation. The response is more sensitive in Cordero as compared to the strain Temecula 1. So biofilm is the main virulence factor through which the Xylella fastidiosa clogs the vascular bundle. So we observed a very distinct pattern in both the strains observed. Um, so Temecula 1 showed significant biofilm reduction at highest tested concentration, while whereas Cordero could not show the significant statistical difference at the highest concentration, but the decline in the biofilm formation was a hope-providing message that the highest tested concentration may reduce the tested for the in vivo studies. This is the most interesting observation we found in this study. So when we observed the live cell discrimination at different time points, the principle is simple that this dye can only penetrate the compromised cells. So higher the level of delta CT, higher the cell death. So we can see that in case of different treatments, even the growth is not, even the cell death is not zero, which means the CFUs, colony forming units, were zero at the higher tested concentration by the end of experiment. But here we can see yet there are some cells which are live. So this finding can be inferred as that the CFU could be zero, but delta CT remain moderate at early time points. So it means the membranes are largely intact, but the cells are not potent to multiply and to create the biofilm under the presence of higher concentrations of cellulose nanocrystals. So we can conclude that cellulose nanocrystal induces a bacteriostatic uh, phase through which the bacteria cannot bacteria may not be able to develop the symptoms in vivo. Based on these observations and the phytotoxicity, we found that it's not phytotoxic in the tobacco plants. We moved to the in vivo evaluations, and here you can see what we have observed by the end of the experiment. So the plants in the red boxes are infected with Cordero and Temecula 1 and not treated with anything after 60 days. The basal part shows the significant leaf necrosis in terms of both, in presence of both the strains. However, when we treated with cellulose nanocrystals, and we have done only two um, treatments, 3 and 7 days post inoculation, and left the plants under optimum conditions to see the visual observation. So here are the plants after the 60 days of infection. So, not just visual appearance, we calculated this in terms of area under disease progress curve. And we have seen that over the period of time, the untreated plants infected with Cordero strain, the total disease burden was up to number 60, while when we treated it with cellulose nanocrystals, it reduced up to 8. That is more than 80% disease suppression. And the similar behavior was observed in case of Temecula 1 infection in tobacco plants. So we further evaluated the symptoms and cross-checked with them with the bacterial load at three different stem points, as I told earlier, below the infection point, above infection point, and at the top of the plant. So the bacterial spread was reduced at all the 3 points, and which further validates our nanobarrier hypothesis. And significantly in Temecula 1, the bacterial load was significantly different at all the three observed plants. However, Cordero, which is known to be very persistent and notorious for its survival and survival ability, we also observed a significant reduction of bacterial load in the top part of the tobacco plants. So, other than the visual observation and bacterial quantification, which was done at two specific time points in the middle at the end of points, we observed the plant behavior using the dual X handheld meter and the polypen device for seeing the pre-symptomatic and post-symptomatic efficacy in terms of different vegetal indices. So in this figure, we can see that the chlorophyll formation. The green line represents the plants which have been treated with cellulose nanocrystals, while the orange line shows the plants which are infected but not treated. In terms of both the Cordero and Temecula 1, we observed that after an initial decline of the chlorophyll, the plant rescued themselves, and the photosynthetic machinery may have adopted the condition with the help of cellulose nanocrystal, because the bacteria was not able to further spread into the plant, and the chlorophyll formation was significantly improved throughout the period of time when the plants approached the end of experiment. So we observed we calculated more than 70 vegetation indices physiological rescue in terms of different indices that we are going to see in next some slides. The first of all, we have photochemical reflective index. It represents the vascular water status of the plants. So we can see the orange lines, which are the infected non-treated plants. It reduced significantly by the end of experiment. So the experiment showed that initially the plant stress was silent, but uh, after 45 days of infection, the stress appeared to be enhanced significantly. However, when the plants were treated with cellulose nanocrystals, they maintained a um, healthy pattern of different vegetation indices, which is confirmed through PRI, optimized soil adjusted vegetation indices, also the indices related to leaf scorch and canopy collapse, and the Carter stress index. So here we have a very distinct um, uh, evaluation of the vascular rescue and canopy integrity. So this plot represents the Cordero plant. So after almost the 45 days of infection, the xanthophyll collapsed in uninfected, untreated plants, while the infected and CNC treated plants maintained a healthy level by the end of experiment. And same was observed in terms of canopy integrity for the Temecula 1 plant, one experiment, that the infected untreated plant were collapsed and during the severe infection phase. However, the infected and treated plants maintained a healthy level of this vegetation indices. So this modified soil adjustment vegetation indices, it shows that the chlorophyll chlorosis was the conventional chlorosis related vegetation indices shows that the chlorosis was suppressed and the plant maintained the chlorophyll status throughout by the end of infection period. So there is a significant difference between the optimized soil adjusted vegetation indices at the end of experiment between infected untreated plants and infected and CNC treated plant. Similarly, for the Cordero, we observed a significant pattern of the plant's better health condition by the end of experiment when we use the PCA analysis to form different hulls, the uh, most of the significance laid on the principal component one, we can see that the brown or orange hulls, which are infected, untreated plants, they were distinct from the blue hulls that are the treated plants. Same for the Cordero. Temecula one, we also observed a clear distinction of orange and the blue hull. And also the global pattern shows that both strains, the untreated negative hull was different than the treated positive whole. So this also verifies our hypothesis that the cellulose nanocrystals, irrespective of their biochemical interaction, they may physically and based on the electrostatic charge on their surface, they may help the plant to escape the stress caused by the Xylella fastidiosa inside the tissues. So the hierarchical clustering, the cluster analysis, it showed that the CNC treated plants were clustered with the healthy plants, and the spectral fingerprint, it represents that our tested level of cellulose nanocrystals were not phytotoxic, and it is effective against in terms of different vegetation indices, it is effective against the stress caused by the Xylella fastidiosa, not only in Cordero, Temecula, but universally both strains show the same behavior. So based on all these observations from in vitro to in vivo evaluation, we propose that cellulose nanocrystals have a promising interaction pathway against Xylella fastidiosa. That is, if we precisely deliver the cellulose nanocrystals suspension into the xylem, it may bacterial growth, which can suppress the bacterial growth and limit the biofilm production. So ultimately, it may prevent the plants from vascular blockade and help the plants to prevent the symptoms of the in different phenotypic parameters. Although this model is mechanistically supported, but our work is still going on into the Puglia region for accessing the feasibility into the almond plants. And similarly, we are observing further elucidate this pathway in almond plants at Institute of Sustainable Agriculture in Córdoba. So the most important question about Xylella fastidiosa with respect to if we only talk about olives, and this data and these numbers are based on the EFSA reports that the cost per tree could be up to 150 euros, but there is more than the cost only. There is a buffer zone that we have to maintain, and then there is the tree replanting timeline, which takes decade decades of the period to replace the plant to the back to that optimum production state. So the CNC endotherapy could be a competitive candidate here because this
I
is
prepare something with no Coca-Cola. Okay. So,
CNC endotherapy could be a
Non
cost
so,
effective
ha parlato una
and
Linda, pensavo che
smart. Sorry. So this is a zero residual material, and it is produced from the wood pulp. The dose effective dose is very minimal, and even the low-tech equipment can be used for the endotherapy purpose. And we propose that plants may be saved from the eradication if we further have the evidence of the based on our ongoing studies, if we get the further evidence of the efficacy of cellulose and cellulose nanocrystal endotherapy, it could be a promising solution for this enormous problem that we have been seeing in all the parts of the world and many different hosts. So that was about my findings, and this study as a whole, it tells us that cellulose nanocrystal is potent for suppressing the Xylella growth under in vitro conditions. It disrupts the biofilm formation, that is very important virulence factor of this bacteria. It blocks the translocation across the acropectal parts of the plant. And it induces a viable but non-culturable state, which is a very novel mechanism observed here, that despite the non-culturability and despite the visual suppression of the bacteria, they may be live, but in presence of cellulose nanocrystal, they may not be able to produce biofilm and clog the plant vessels. So in this study, we have observed up to 87% of disease reduction in vitro conditions in tobacco plants in case of Cordero strain, which is a most notoriously known strain so far. And the spectral phenotyping proof shows that our treatment is non-phytotoxic. It helps the plant to rescue the physiological machinery, and all the vegetation indices shows that at this therapeutic level, it is non-phytotoxic, and it helps the plant to prevent the Xylella fastidiosa disease symptoms. So right now, our one experiment is going on to further elucidate this experiment in almond plants for the subspecies multiplex, and also we are doing the experiments in the field at the natural infected field level in Apulia region against the Pauca subspecies. So our ultimate goal is to translate to olive and almond endotherapy based on the further observations. I would like to express my cordial thanks to the team of Dr. Blanca at Institute of Sustainable Agriculture. They hosted me for almost 1 year, where I have performed all these experiments, and still we are in collaboration. And I would like to thank the University of Tuscia, which have been providing me support throughout. And thank you so much, all of you, for hearing me. And if you have any questions, you're most welcome.
Thank you, Amir. We were really happy to have you here. You have been working very, very hard, as you have demonstrated. That show with only a small part of all the experiments you have performed with us. So does anyone have some questions for Amir?